What do you get when you mix 1 part bacterium, 5 parts virus, a protein, an RNA, and ingenuity? By the end of this video, you'll know the answer to that question and more. We're going to take a look at the role of microorganisms in one of the hottest career sectors in Massachusetts, biotechnology. By the end of this video, you should be able to explain what biotechnology is, give a definition, and you should be able to explain how microorganisms are involved in recombinant DNA technology, gene therapy, and CRISPR. Simply stated, biotechnology is the manipulation of living organisms or their components, things like proteins, lipids, and DNAs, to produce useful products. The science of biotechnology has existed for centuries. Archaeologists have found that humans have been using bacteria and fungi to ferment breads, yeasts, and rice for over 8,000 years. We also know that over the centuries, farmers have used selective breeding to develop the corn that we eat today from ancient grasses. They've also bred animals like cows in specific ways to develop breeds that produce lots of milk or pack on extra muscle, both of which bring more money at the market. While biotechnology is still used in agriculture today, it's more commonly used to help develop crops that are drought or pest resistant. It's also used to develop animal breeds that secrete new vitamins and proteins into their milk. This kind of biotechnology, modern biotechnology, is a multidisciplinary field, and it overlaps many studies such as biology, biochemistry, chemistry, physics, mathematics, genetics, and statistics. Typically, when someone says the word biotech, however, images of DNA, proteins, and mutant animals or plants come to mind. I would call that modern biotech, and much of that work is the result of major advances in the discipline of molecular biology. Molecular biology attempts to explain cellular processes by studying the relationships between DNA genes and the RNAs and proteins produced from them. So this slide just lists some of the important discoveries that underpin our knowledge of molecular biology. I wouldn't expect you to remember these dates and events for an exam, but they are useful to know just for your own information. Recombinant DNA technology was birthed out of the field of molecular biology. In 1960, Paul Berg, an American scientist, figured out that you could take a piece of DNA from an animal and insert it into the DNA from a bacterium. Then, if you put this hybrid DNA back into a bacterium and stimulated the bacterium properly, you could get it to make an animal protein from that animal DNA. This hybrid DNA was called a recombinant. Recombinant DNAs are DNAs that are made from two different sources, in this example, an animal and a bacterium. One of the easiest ways that scientists can make a recombinant DNA is by using plasmids. Plasmids are circular extrachromosomal DNAs, and they're bacterial in origin, oftentimes. So if this is a bacterium, every bacterium has a central chromosome, and this is usually a circular piece of DNA. It has all of the essential genes for the bacterium, and it's looped in an area called the nucleoid. Plasmids are DNAs that are not part of that big loop, and they can exist in many different locations. So they are extrachromosomal because they're not part of this central chromosome. Typically, plasmids are small. They house fewer than 30 genes, and the genes that are on a plasmid are usually non-essential, but they may help increase bacterial survival under certain circumstances. So for example, if you're a bacterium and you're in an environment where antibiotics are present, but you have a plasmid with an antibiotic resistance gene, then you'll be able to survive in the presence of the antibiotic, whereas a bacterium that doesn't have that plasmid would die. Plasmids can exist in the cytoplasm of the bacterium, like I drew here, but they could also be removed from the cytoplasm. Sometimes these plasmids get incorporated into the central chromosome of the cell, in which case they would be called episomes. So let me tell you a quick story to help you understand the usefulness of recombinant DNA technology. So by now you should have had AMP2, and you're probably familiar with the disease diabetes mellitus. Diabetes mellitus is a disease in which the pancreas doesn't make enough insulin for good health. Remember, insulin is this hormone that binds receptors on body cells and causes those body cells to absorb glucose, okay, from the blood. That glucose goes into the cell makes ATP from it, and then it's able to carry out chemical reactions and other cellular functions. This lowers the amount of glucose in the blood. So someone who has diabetes oftentimes measures their blood glucose level, and that tells them something about how their cells are absorbing glucose. They may take an insulin shot daily to stimulate their body cells to take in glucose. And they may do this around mealtime. Okay? So humans have known about diabetes for centuries. We know that doctors in ancient Greece and China and Egypt and India and Persia and we know that doctors in the Middle Ages did this as well. They would taste the urine of their patients, and those who had diabetes would have very, very sweet tasting urine. Treatment at this time, and for centuries afterwards, consisted of severely restricting the diets of diabetes, sometimes to starvation, in order to keep the blood glucose level low and extend someone's life. It wasn't until 1889 that two scientists, doctors, Joseph von Mering and Oskar Minkowski, discovered that when the pancreas is removed from dogs, the dogs developed the signs and symptoms of diabetes. This is the first time that the pancreas was connected with diabetes. It wasn't until 1910 that doctors figured out that it's insulin that the pancreas is making that's lacking, okay, in people who have diabetes. So people who have diabetes don't make enough insulin, in other words. It wasn't until 1921 that someone actually shows that insulin, or lack thereof, is responsible for diabetes. So 1921, Sir Frederick Banting and Charles Herbert show that if you extract insulin from cows and give that to diabetic dogs, then it reverses the diabetes in the dogs. The first human treatment for diabetes came in 1922 when Leonard Thompson, a 14-year-old boy who was basically at death's door, was injected with insulin and insulin extract, and it turned out to save his life. So for decades, the world treated diabetes by sacrificing pigs and cows to recover their pancreases and extract insulin from them. In 1978, a scientist from Genentech named Dennis Clyde toured a plant in Indiana that was making insulin from animal pancreas. He noted that it took 8,000 pounds of pancreas glands from 23,500 animals to make 1 pound of insulin. Eli Lilly was the major insulin producer at the time, and they said that they needed something like 56 million animals to make enough drug to meet the demand in the United States. This was in 1978. So scientists at Genentech, under the leadership of Clyde, used recombinant DNA technology to produce insulin in bacteria. Let's get an idea of how they did that. So in recombinant DNA technology, a foreign DNA, let's say it's human insulin, is attached to a bacterial DNA. So in other words, we would construct a human insulin gene and we would want to attach it to a bacterial DNA. And this bacterial DNA is the plasmid that I was talking about a couple of slides ago. This new DNA that consists of the human gene and the bacterial DNA, that's called a recombinant DNA. So the DNA is coming from two sources. When that recombinant DNA is added to a bacterium, and then you stimulate the bacterium to make protein from the DNA, the bacterium is going to make protein from all of the bacterial DNA as well as from the human gene that was inserted, because the bacterium doesn't know the difference between what's a bacterial DNA and what's a human DNA. Once you have all of that human protein inside of the bacterial cell, you cut the cell open and you separate out the human protein from the bacterial proteins. In this case, you get human insulin. In a little bit more detail, we're going to take a plasmid DNA and we're going to take our human insulin gene rather. We're going to cut it with an enzyme called a restriction enzyme. So restriction enzymes are actually extracted from bacteria. If you're a bacterium, one of the worst things that can happen to you is you get infected by a virus, because most bacterial viruses kill bacteria as part of their life cycle. So bacteria have developed these enzymes that recognize sequences that are on viral DNAs and then cut those viral DNAs, which interrupts the virus life cycle. Scientists have kind of figured out what these enzymes are. They've extracted them, and now you can buy them in a catalog. So these enzymes, again, are called restriction enzymes, and we can use those enzymes to cut the plasmid DNA and the human DNA. This makes the ends of the DNA, when I say the ends, it's this part here and here. It makes the ends of the DNA match, meaning the nucleotide sequences on those ends kind of fit together like puzzle pieces. If we add another enzyme called ligase, which is like a molecular glue, we can actually join the human DNA to the plasmid DNA, making one piece of DNA, which is the recombinant DNA. Then, through a process called transformation, we weaken the bacterial cell and cause it to take up that recombinant DNA. So this rDNA that you see here, that's the recombinant. Then we stimulate the bacterium to produce the insulin, and we cut the bacterium open and extract the insulin protein. And this technology is used to produce a wide variety of therapeutic drugs. It's used for producing some of the proteins that you find in laundry detergents. So you know that laundry detergent that breaks down grease stains that you might get on your clothing when a hamburger drops onto your shirt. Bacterial proteins called lipases break down the fat from that stain and help clean your clothes. You need a lot of lipase to do that, so recombinant DNA technology is used to produce large amounts of lipase for detergent. We use recombinant DNA technology to produce vaccines. You might be familiar with Gardasil. This is the vaccine that protects against 3 or 4 different types of human papillomavirus. These are viruses that are responsible for cervical cancer. And so we use bacteria to produce little pieces of the human papillomavirus, and then we inject those pieces into someone's body. The immune system reacts to those pieces and learns how to recognize them, so that if someone becomes exposed to human papillomavirus later on, the immune system already knows what that virus looks like, and it can defend the body against infection. I mentioned producing insulin using recombinant DNA technology. Humulin is an example of a bacterially produced insulin. You can see here on the box it says rDNA origin. Procrit is erythropoietin. It's an enzyme that stimulates the production of red blood cells. So it's produced using recombinant DNA technology. This is Herceptin, which is an anti-cancer drug. It's also produced using recombinant DNA technology. Another biotechnology that uses microorganisms is gene therapy. Gene therapy uses viruses, like this one, to introduce new DNA to cells that compensate for missing or defective genes. In theory, this should restore normal function to a cell. We know that genes that are just injected into a cell directly, they don't function properly. So instead, viruses are genetically altered, like you see here. So we have some viral DNA. We cut parts of the viral DNA out and insert our new gene in. Then we package that into a capsid and introduce that virus to cells. The virus is brought into the cell. Many viruses actually have part of their life cycle inside the nucleus. So the DNA that's inside the virus is removed from the capsid protein of the virus. This releases naked DNA. And then that naked DNA is translocated or transferred to the nucleus, where it can be incorporated into the chromosome of the cell, or it can just exist in the nucleus. Either way, the cell will transcribe the gene of interest here, and those RNAs go out to the cytoplasm where they get translated to make protein, and this restores function, in theory. There are some technical difficulties, however. For example, we don't have all the right viral vectors that are necessary to target all the different types of cells in the body. So that's one problem. Another problem is we need to be able to control which tissues get infected. Right now, we don't really have a good way of doing that. Another problem is that sometimes the body reacts to the virus and you get an extremely severe inflammation response. Sometimes that inflammation response leads to organ failure, and in the worst cases, it leads to death. Another technical difficulty is controlling the expression of the genes in the body. So there's a lot of things that have to be worked out in order for gene therapy to be a more robust therapy, but it has been used to treat some cases, some symptoms of cystic fibrosis, some blood and marrow cancers. It's been used to treat Duchenne's muscular dystrophy, and then severe combined immunodeficiency disease. So this last one, severe combined immunodeficiency disease, is called the boy in the bubble disease. Basically, it's a very rare genetic disorder where the immune system doesn't function normally. It doesn't protect the body against infection by common garden variety bacteria. So people who have this disease are highly susceptible to infection and death from microbes that for most of the rest of us wouldn't cause a problem. They're extremely sensitive to microbes. For this reason, people with this disorder often have to live in kind of isolated environments. This is David Vetter, and he was outfitted with a spacesuit that basically provides a sterile environment inside the suit and isolates him from the microorganisms in the environment outside of the suit. So let's see what you remember. Just a quick check. Which of the following is an example of biotechnology? Would it be using penicillium molds to ripen cheese, using E. coli to produce enzymes for laundry detergent, or using adenovirus to deliver functional cystic fibrosis transmembrane conductance regulator genes to deficient cells? You might want to pause the video and then press play when you have your answer. So let's see what you remember. Just a quick check. Which of the following is an example of biotechnology? Would it be using penicillium molds to ripen cheese, using E. coli to produce enzymes for laundry detergent, or using adenovirus to deliver functional cystic fibrosis transmembrane conductance regulator genes to deficient cells? You might want to pause the video and then press play when you have your answer. Here's another one. A recombinant DNA is that a foreign gene introduced to a cell using a virus, a DNA made from two sources, or only made from human DNA? Right. A recombinant DNA, no matter what it is a DNA made from two sources. So in my example, I gave you insulin, right? Where we had a human gene inserted into a bacterial uh, DNA. You could have a fungal gene inserted into a bacterial DNA. You could have a viral gene injected into a fungal DNA. You could have a DNA that's made from two different parents. For example, mom's giving some genes from her chromosome and dad's giving some genes from his chromosome. You probably remember crossing over from your Gen Bio class. Well, crossing over is an example of a recombinant DNA that crossing over results in a DNA from two sources. What about plasmids? Are plasmids pieces of chromosomal DNA? Or do plasmids contain essential genes only? Or do plasmids potentially contain antibiotic resistance genes? What do you think? Right. Plasmids may contain antibiotic resistance genes. Remember, plasmids have non-essential genes that may aid the survival of a bacterium only in certain circumstances. Remember, these are extra chromosomal, so they're not chromosomal DNAs. At the very beginning of this video, I asked you, what do you get when you mix 1 part bacterium, 5 parts virus, a protein, an RNA, and ingenuity? The answer is CRISPR. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. Remember a few slides ago, I said that one of the worst things that can happen to you if you're a bacterium is that you get infected with a virus because bacterial viruses often kill bacteria? Well, back in 1987, scientists at Osaka University reported finding weird DNA sequences in bacteria. They found 5 DNA sequences that were identical, which is unusual in bacteria, but they were separated by short stretches of different sequences. Over time, Scientists found that lots of bacteria had these sequences, and eventually scientists matched the stretches of different sequences to viruses. That's right, the bits of DNA between the repeating DNAs were bits of viral DNA. We now know that when a bacterium gets infected by a virus, if it happens to survive, it cuts and stores that viral DNA. It's like a mugshot, so the bacterium can remember the invader. If the bacterium is infected with that virus again, it makes an RNA copy of the mugshot, which will then bind to the DNA of the invading virus. An enzyme called Cas9 will bind the RNA as a guide and cut the viral DNA. This stops the viral infection. It's like an immune system. As usual, scientists have figured out how to do something useful with this knowledge. They use CRISPR-Cas9 to quickly and cheaply change an organism's DNA. You can think of this system as being a genome editing tool. You might remember back in 2018, a Chinese scientist used this technology to change the DNA of a couple of Chinese infants to help them keep from getting infected with human immunodeficiency virus, or HIV. I'll share a podcast with you later in this semester that talks about CRISPR-Cas9 in more detail. So that brings us to the end of this video. I've defined the term biotechnology. Remember, it's the use of living organisms or their components to produce useful products. Further, we've talked about how microorganisms are involved in biotechnology. Specifically, we looked at 3 tools or techniques. We looked at recombinant DNA technology. This is one in which a DNA is made from 2 different sources. So remember, we talked about the human insulin gene as an example. The human insulin gene was inserted into a bacterial plasmid. Then that recombinant DNA was put into a bacterium, and the bacterium was stimulated to produce insulin. That insulin could then be extracted and purified for use in therapeutics. This technology has undoubtedly saved millions of lives, both human and animal. We also talked about gene therapy, in which a missing or defective gene is replaced by a gene that's carried by a virus. When a virus infects a cell, then that normal gene that's in the virus gets transferred to the cell. Then we talked about CRISPR. Remember, CRISPR is a technology that was derived from bacteria, in which a mugshot from a viral DNA, from a virus, is used to protect a bacterium from reinfection by the same virus. Scientists nowadays use this technology to modify the genomes of other organisms. In my next video, we're going to talk a bit about the role of microorganisms in human health. I look forward to seeing you there.